So our next speaker is Professor Jian Feng Ma. So he's a professor, leader, the head of the Institute of Plant Science and Resources at Okayama University. And he's had an amazingly productive career in plant mineral nutrition, made a number of discoveries, identifying a number of aluminum tolerance genes in different crop species and their mechanisms. It's shown that silicon, he cloned the first silicon transporters, showing that really the significance of silicon as a beneficial element. Selenate transporters, arsenic transporters. He's done a lot of work on manganese and other micronutrient nutrition, which he's, I think, going to talk about some of that today. He's, again, another one of the highly cited researchers and is published in the top journals like Science and Nature. So I think today we're going to hear about the work he's doing, really interesting work in the nodes, the rice nodes. And again, this is vascular biology. It's the interplay of the xylem and phloem in regulating, I believe we're going to talk about mineral nutrition and micronutrient nutrition. So Jian Feng, please come on.
So thank you, Leon, for your very, very kind introduction and also for the invitation. As many speakers mentioned, the plant only requires 14 essential elements. These elements usually present in the soil, and they will be taken up by the roots, and then translocated from the root to the shoot, and finally will be derived to different tissues, and the organs depend on the requirements. And on the other hand, in soil, in addition to this essential element, also have toxic elements such as cadmium, arsenic, aluminum. So these elements will also be transported into the grain or edible parts, and it will affect our health. Now we know that all this process, they need a lot of different transporters. Therefore, understanding transport is very important for the better and safe crop production. I will not talk about the transport involved uptake, and I will focus on the transport involved in the distribution, especially to the distribution to the development tissues such as young leaf and the reproductive stage organs. As I mentioned, that after the uptake of mineral elements, they will be transported to the top part with transpiration. So please imagine if distribution follows transpiration, more minerals will go to the older leaves because they have the higher transpiration rate. But actually, development tissues such as young leaf, reproductive organs, they require they have very low transpiration rate, but they require higher mineral elements. So therefore, this means the plant have a system to control the mineral element distribution. But for a long time, we don't know how the mineral elements are derived to the developing tissues, which has a very low transpiration rate. Next one, please. So recently we found that in gramineous plants, nodes plays a very important role in the mineral element distribution. So today I will tell you 3 different examples to tell you how these elements are preferentially derived to the developing tissues, especially to the grain. Before I go to tell you the detail of the transporters involved in the distribution, preferential distribution developed tissues, I just want to give you a very brief introduction about the nodes. Next one, please. So node, yeah, I think you can see like this in the gramineous plants such as rice, barley, and wheat. The next one, please. So in modern rice cultivar, actually there are 13 to 18 nodes, but you cannot see them at the vegetative stage because they stuck together at the bottom. Only at the reproductive stage, the up 4 to 5 internode, they start to elongate, and then you can see the node like this. So when you count the nodes, we usually count from the top, node 1, node 3, but not from the from bottom because you cannot discriminate them very clearly. Next one, please. So actually, the node has a very complicated but very well organized vascular systems. There are two major vascular bundles. Next one, please. One is we call the enlarged vascular bundle. It connected to the roots and the lower nodes, and then expanded to the leaf. Next one, please. Another one is we call the diffuse vascular bundle. It initiated in the nodes and then connected to the developed tissues, such as the young leaf and the grains. Next one, please. Therefore, when mineral elements taken by the roots are translocated through the transplantation rate via enlarged vascular bundle, they must pass this mineral element to the diffuse vascular bundle to reach the developing tissues. We call this process intervascular transfer. Okay? Intervascular transfer. So actually, there are many transporters required for the intervascular transfer. Due to limited time, I will not tell you more about the nodes. If you are interested in nodes, we have a video abstract for the trends in plant science. Next one, please. You just go to YouTube and type, next one, please, Ma node, and you will find me. Next one, please. Just want to remind you, never mistype Ma nude, okay? Next one, please. So to find transporters expressed in the node and to find the transporters response for the distribution of mineral elements, we combine the laser microdissection with microarray. Recently, we used the RNA-seq to cut out different tissues, such as phloem of enlarged vascular bundle, xylem of enlarged vascular bundle, and the diffuse vascular bundle, and then do the microarray or RNA-seq analysis and try to find some transporters highly expressed in different tissues. So today I will tell you 3 different examples to tell you how these transporters are involved in the preferential distribution of mineral elements. Next one, please. This is a summary of transporters we identified in the nodes. Okay, next one, please. The first one is my favorite element, silicon. Okay, I will give you a very brief introduction about the silicon. This is a beneficial element, and next one, please. And it is the most abundant mineral, actually, in the Earth class. Next one, please. So actually, it's an essential element for animals. So this was demonstrated in 1972. They used the chicken, as you can see here. If they give the silicon to chicken, they grow very well. But without the silicon, you can see this chicken become very smaller. And they found that the silicon is involved in bone information. So when I show this picture to students, they always ask me, what happened to us if we don't take enough silicon? It's very difficult to test in human because, silicon is the most abundant elements in the Earth clusters. You cannot completely remove silicon from the food and from the drinks. So my imagination is, next one, please. Like this, okay? So this, he become very famous due to this slide. He's my PhD student, but you don't need to worry about this. Okay, next one, please. Next one, please. Because if you drink beer, next one, please, you will get immediately will get the recovery. This is because, next one, beer is very rich in silicon, okay? So I analyzed beer silicon concentration in Japanese beer, about 30 different beer. You can see some beer contain very high silicon, about 60 ppm. Some beer only contain 10 ppm. The interesting thing is there's a good correlation between price and the silicon concentration in beer. Expensive beer contain more silicon, in the cheap beer contain less silicon. Okay, this is true. Anyway, this is my unpublished data. Next one, please. Okay, so a newspaper said that beer builds strong bonds because beer is rich in the mineral elements. So if you want to have a strong bond, especially young people, okay, not beer, beer locus, okay, you need to drink expensive beer, okay. The next one, but for plants, it has not been recognized as an essential element. It's a beneficial element. Next one, please. So next one, please. Silicon actually has a lot of beneficial effects on plant growth and development. But the important lure is it can enhance the tolerance to different stress, including abiotic and biotic stress. It is especially very important for the higher yield, higher and sustainable yield of rice, because rice is silicon accumulating plants. As you can see here, if you have a high silicon, it will more resistant to the disease, this is rice blast, to the insect, and also it's more resistant to the typhoon, okay? When this picture was taken at the harvest, you can see with high silicon, the green shows the golden yellow color, and also the panicle is bended. But with low silicon, you can see that the panicle is very due to the infection of different pathogens, and also it is elected due to the low fertility. So silicon is very important for rice production. So as I mentioned, it has not been recognized as an essential element in the viewpoint of plant physiology, but it is very important for rice production. So in Japan, we call the silicon as agronomically essential element. So every year they apply silicated fertilizer to paddy field to increase the rice yield. Okay, next one. So many years ago, as Leon mentioned, we tried to understand why rice is able to accumulate such a high silicon concentration in the shoot, also in the grain, especially in the husk, and we found two different transporters, ELISA1 and ELISA2, and ELISA1 is an influx transporter, ELISA2 is an efflux transporter. They are polarly localized at the exodermis and endodermis. As you can see here, ELISA1 is localized as the green color is localized at the outside. There's a special name. Proximal side and another one. What is another one? The steel side. Okay, thank you very much. The steel side and ELISA2 is localized at the proximal side of the stem cell. Okay, they are very important for silicon uptake. But today I will not talk about these two transporters. I want to next one, please. Tell you how silicon is preferentially derived to the panicles, because the high silicon accumulation in the panicle, especially in husk, is very important for the higher yield of rice. As you can see here, at the harvest, the husk accumulated about 8%. Okay? It is many times higher compared to the nitrogen, phosphorus, and potassium. But the husk, does not have stomata, and also the surface is much smaller compared to the flag leaf. So this means the transpiration is very low. Why silicon is able to be highly accumulated in the husk? So we found that the transporter in the node play a very important role. So we found that we analyzed the expression profile of several silicon transporters, and we found that ELISA2, ELISA3, and ELISA6. ELISA6 is a homolog of ELISA1, and ELISA3 is a homolog of ELISA2. They are efflux silicon transport, and this is ELISA6 is an influx transporter. They are very highly expressed in the nodes. Next one, please. So where ELISA6 expressed, and we found that it is expressed in the xylem transport cells, as you can see here. Here's a phloem, and this is xylem, and it is a specific cell we call the xylem transfer cell. You can see the plasma membrane is folded due to the ingrowth of cell wall. So this is to increase the uptake surface. You also can see that this transporter show a polar localization facing to the xylem. So when you knock out this gene, what happened? So we analyzed the silicon concentration in different tissues. As you can see here, next one please. If you knock out the ELISA6, you got increased silicon concentration in the flag leaf. Next one, please. But the decreased silicon concentration in the panicle parts. Next one, please. So what about the ELISA2? And we did a double immunostaining, and we found that the ELISA2 is a pink color, and ELISA6 is a green color. You can see ELISA2 is localized at the cell just beside the ELISA6. It also shows the polar localization. Next one, please. So how about ELISA6, ELISA3? Okay, we also did a double immunostaining using specific antibody for ELISA3 and ELISA6. As you can see here, this is ELISA6, and this is ELISA3. And actually, you cannot see here, this is ELISA2. Okay? So you can see that the ELISA3, so what happened if you look at ELISA2, ELISA3? So we did a stem feed experiment. We cut the panicles and then fed silicon from the cutting end and look at the distribution of silicon to the flag leaf or to the panicles. As you can see here, similar to ELISA6, when you look at ELISA2, ELISA3, you got increased accumulation into the panicles, into the flag leaf, but decreased to the panicles. So this means that all these three transports are involved in the distribution of silicon to the panicles. Next one, please. And then we did mathematical modeling, okay, using three transporters, and try to understand what are other factors are involved in the silicon distribution. And as a result, we found that in addition to these three transporters localized at the different cell layer, ELISA6 here, ELISA2 here, and ELISA3 here, other two factors are very important. One is enlargement of the nodes. Okay, this will slow down the transportation rate and let the mineral elements stay longer in the node. Okay, the another one is we found in the node actually there's a apoplastic barrier. In the roots, you know, there are Casparian bands, but we found that in the root, in the node, there's a Casparian band, similar to Casparian band. There's an apoplastic barrier at this cell layer where ELISA2 are localized. This is also very important to prevent the return of mineral elements from the diffuse vascular bundle to enlarged vascular because the transpiration pressure is very high in this side. Okay. Next one, please. Okay, so now I will move to second example about phosphorus. As Hong Liao mentioned, this phosphorus is very important for the crop production. So to increase crop production, you need to use fertilizer. Okay, so every year you need to fertilize. And as she mentioned, not he, okay, as she mentioned that actually the efficiency of phosphorus fertilizer, use efficiency of phosphorus fertilizer is very low. Or maybe maximum is 20%. Okay, but this result. Next one, please. High accumulation of phosphorus in the grain. This is not good actually, because about 60 to 85% of total phosphorus is finally allocated to grain. Usually, phosphorus in the grain is important for the germination in the early growth, but only 1 milligram phosphorus per gram is necessary. However, in the modern agriculture, usually the grain produced contain more than 2 milligram per gram, and in our university field, usually we got 4 to 5 milligram per gram. So what is the negative effect of such higher phosphorus in grain? Actually, there are three negative effects. One is, next one please, it will remove of phosphorus from soil. Second one, please, it will reduce the bioavailability of essential metals such as iron and zinc. And the third one, please, it will also increase the risk of eutrophication of waterways. Why? Next one, please. Because in the grain, about 65 to 80% total phosphorus is present in the form of phytic acid. Next one, please. As you can see here from this structure of phytic acid, it contains a lot of 6-phosphate group. Next one, please. But we cannot digest phytate. And not we, and most animals also cannot digest this form of phosphorus, phytate. Next one, please. So this means, next one, please. When we eat grain or animal eat grain with a high phytate, because we cannot digest it, it will be released to the environment. So this will cause eutrophication of waterways. And also because it has a strong binding capacity with the zinc and iron, so it will decrease the bioavailability of zinc and iron. So next one, please. So for many years, people think that reducing grain phosphorus is a very important issue for sustainable agriculture and environment. Next one, please. So we want to know what is the source of grain phosphorus. Next one, please. Actually, there are two different sources. One is retranslocation of phosphorus from older leaves. Next one. And the second one is node-based distribution of phosphorus newly taking up after flowering stage. So we focus on the second one. Next one, please. And as I mentioned earlier, we use the laser microdissection combined with microarray, and we found that the one gene annotated to OS-TR34 is highly explicit in the nodes. So next one, please. So next one, please. So this gene actually belongs to the sulfate transport family, and so far in Arabidopsis, some members were reported to be low affinity or high affinity sulfate transport. But we found that SALTR34 is not sulfate transport. This is a phosphate transport. Next one, please. We use different approach to demonstrate that this is a phosphate transport. Next one, please. So this is just to show you one example. We use in vitro method and purify the protein, this protein, SALTR34, and we renamed it to SPDT. I will tell you the reason later. And then put in the liposome and did a transport assay. As you can see here, when you have this protein in the liposome, you got the transport activity for phosphate. When you add 5 millimolar cold phosphate, you got the decreased transport activity because they compete each other. However, when you add 5 millimolar sulfate, you cannot see any inhibition. So this means that this is a transport of phosphate, not for the sulfate. Next one, please. So based on this result, we change the name. So this is actually not a sulfate transporter, it's a phosphate transporter. So we change the name to SPDT, SALTR like phosphate distribution transporter, SPDT. Next one, please. This is because based on the single pore double slow SPDT, because it's functioning like a switch, okay? Next one, please. Like a switch, it switched to the green part or switched to the next one, please. And then we investigated the expression pattern in the field. As you can see here, this is just to show you the expression pattern at the reproductive stage. You can see it is very, very highly expressed in the node 1. And we also investigate the tisular and the cellular localization. As you can see here, different from the ELISA 6 and the silicon transport I mentioned earlier, it is localized in the xylem region of enlarged vascular bundle and the diffuse vascular bundle, but not in the phloem region. And this is just to show you that it is nicely localized at the plasma membrane. So it is a plasma membrane localized transport for phosphate. Next one, please. So what happens if you knock out this gene? So we did a lot of actually phenotypic analysis using the 3 different mutants. And this is just to show you one result. You can see that under the phospholimited condition, we found that the youngest leaf, you can see the growth was seriously inhibited. But if you compare the phosphate uptake, you can see that there's no significant difference in the phosphate accumulation between the wild type and the mutants. However, next one, please. If you look at the distribution in the different tissues, you can see that knockout of this gene resulted in decreased phosphate distribution to the short basal region. This included nodes, but the increased the distribution to the older leaf, but the decreased the distribution to the young leaf. Okay, next. This means that if you look at the SPD, it does not affect the phosphate uptake, but it affected the phosphate distribution, especially to the young leaf. developed tissues. Next one, please. And at the reproductive stage, we found that similar result. Knockout of this gene, it resulted in increased accumulation in the older tissues, but decreased in the grains. So this is a distribution rate in different, in the grain and in the straw. In the wild type, about 64% phosphorus is finally derived to the grain. But in the mutant, you can see 42 to 44 of total phosphorus is derived to the grain. So there's a 20% decrease. Okay? So this means that if you look at this gene, 20% of phosphorus was derived to the grain was decreased. Next one, please. And the important thing is, usually if you look at the genes related to phytid synthesis, you will get the penalty of yield. But we found that if you look at this gene, there's no significant decrease in the yield, and also there's no significant decrease in the quality. If you look at this picture, this wild type, and this 3 different mutants, they look similarly. Okay? Next one, please. Most important thing is, if you analyze the phytid in the grain, we found that about 20 to 30% of the phytid was decreased in the mutants. Next one, please. And another important thing is, even if the phytid concentration in the grain was decreased, but it does not affect the germination and also does not affect the early growth. You can see here, the 3 mutants does not show any difference in the germination and early growth compared to the wild type. Next one, please. So based on this result, we think that SPDT localized at the xylem region of the enlarged vascular bundle and diffuse vascular bundle are very important for unloading phosphorus in the xylem of enlarged vascular bundle. So it is also very important for the preferential distribution of phosphorus to the grain, especially under the phosphorus limited condition. But under the phosphorus sufficient condition, this is a very good treat for the agriculture because it can decrease the phytid concentration in grain. Next one, please. So this is a cartoon, just to show you. Next one, please. In the wild type, phosphorus, most phosphorus will be derived to the grain due to the SPDT, but in the mutant, due to the knockout, loss of function of this transporters. Next one, please. Yeah, less phosphorus to the grain, but more phosphorus to the leaves. Next one, please. So this resulted in 20 to 30% decrease in the grain phosphorus. And the next one, please. So have 3 different good things. Once decrease the phosphorus removal from soil, and also decrease the anti-nutrient effect, especially increase bioavailability of iron and zinc, and also it decreases eutrophication of waterways. So this is a very important gene in agriculture, and now this gene, some group in Japan has used this gene for breeding rice with low phytate. Okay, next one, please. So last example is about the toxic element. Next one, please. Arsenic. Okay, next one, please. So arsenic, as you know, is a very toxic element. If you take up arsenic, it will cause a lot of disease, especially skin cancers. Next one, please. Actually, rice is a major source of arsenic intake. Next one, please. Because rice is a staple food for half population of the world, and also rice is able to accumulate high arsenic compared to other crops such as barley and wheat. The reason for higher accumulation of arsenic in rice is one of the major reasons. Next one, please. Is in paddy field, you know, arsenic is presented as arsenite. So arsenite is very similar to silicic acid. So actually, they are taking up through the silicon transport, ELISA1 and ELISA2, as I mentioned earlier. As you know, that ELISA1 and ELISA2 are very highly expressed in roots. So that's why rice is able to accumulate very high arsenic in the grain compared to other crops. Next one, please. So we found that another transporter belong to ABC transport family, OS ABCC1. It is very important for the arsenic distribution to the grain. We found that it is also highly expressed in the node 1 at the reproductive stage. Okay, next one, please. And this is a localization of ABCC1 in the node 1. As you can see here, it is localized at the phloem region. And next one, please. Furthermore, we use different approaches. This is Western, and this is immunostaining. You can see it is localized at the tonoplast, not the plasma membrane. Next one, please. And we also got the mutant and compared the arsenic concentration accumulation in different tissues between wild type and the mutants. And you can see here, next one, please. If you knock out ABCC1, you got decreased arsenic in the node, okay? But increased about 13 times more arsenic in the grain. Okay? Next one, please. And this is a very short term feeding experiment, and this is control. You can see this. We're using the lobigium. There's no difference in the distribution between wild type and the mutants. But in the arsenic accumulation experiment, you can see this is wild type. This is two different mutants. In the wild type, more arsenic is accumulated in nodes, but less to the grain. By contrast, in the mutants, less arsenic is in the node, but more arsenic to the grain. Next one, please. The reason for this is, as I mentioned, that the ABCC1 is localized at the tonoblast of a companion cell, phloem companion cell. So when arsenic is transported to the grain, they must be pass through the phloem companion cell. If ABCC1 is localized, is present here, it will be sequestered into the vacuoles. Then less arsenic will go to the grain. So that's why if you knock out this gene, less arsenic is accumulated in the node, but the more arsenic into the grains. Next one, please. So recently we overexpressed this gene using a specific promoter, and we found that when you overexpress this gene expressed in the node, you got the decreased arsenic concentration in the grain. So this is a very safe way to by manipulating these transporters. Next one, please. So I think this is all what I want to talk. And the final slide, I just want to tell you that transporters are very important for better and safe crop production. We need to understand more about the transporters in different tissues. And this is the last slide. I just want to thank my lab members. And next one, please. And also my collaborator, Yong-Soo Lee from postdoc in South Korea about the arsenic project, and Professor Yoshida in the University of Tokyo for analysis of phytate, and Dr. Miyaji for the in vitro phosphate transporter assay experiment. Next one. Last one. So our findings from GSP. So this is a specially promoted research. Thank you for your attention.